Okay, now I'm online. Okay. Hi, hello everyone. Hello, Microbehunter here, and hello to another live stream. Okay, there's been a little bit of a delay, but I think you should be able to see me now. Video is good, and sound is also good. Cool. Okay, now I'm online. Okay. So I can also hear myself. Yeah, it always takes a little bit of time at the beginning to get everything started. Okay, yeah, hello, welcome again, and welcome to another Saturday live stream. I see that a lot of folks already started to join. Um, yeah, very briefly, what I would like to do today, a little bit based also on due to a request by some of you. Yeah, some of you wanted to have again some live organisms, some observation to again. Yeah, so we're going to do this as well. But I've also prepared a couple of slides here, and I would like to permanently mount those because I would like to compare three different mounting media, which I did not compare before. Right? I mean, I've been making permanent slides already on this live stream before, but at least the mounting medium that I'm going to try today and compare it, I've not done before. Yeah, and that's basically very briefly the plan for today. A little bit of observation, the same specimens that I've prepared already over here, and yeah, we're going to take it from there, right? I would like to use always the first couple of minutes to say hello all around the world. This gives people some time to join in to the live stream. So there is a hello from New Zealand, from Vietnam, from Germany, yeah, from Greece, London, UK, from the Netherlands, yeah, from London, from Germany, Netherlands again, France, the United States, and again the United States. Okay, Belgium, California, yeah, UK. Sound is good, thank you. And Montreal from Canada, Belgium, United States again, and from India even. So it's really nice that people from all around the world are kind of happily united. Yeah, today on a Saturday to do a little bit of microscopy with me. Some of you, many of you already know the format of this stream. You please feel free to ask questions or to comment in the chat. I have to switch over to so-called to live chat. Okay, please feel free to do that. And usually after 1 hour, I will be answering all of those questions that are a little bit off topic. But if you have any questions which are directed to me, please say an @Oliver or @Microbehunter so that it's easier for me to filter out those messages. Okay, so that's a little bit the idea. Yeah, later on the video will of course also be like all time, every time will be online as well. So that means if you, because of time differences, do not want to watch the whole live stream, then do not worry. Everything will be later available also online, provided that there will not be any strange interruption into the live stream like we've unfortunately had before already. So before we start doing this, I want to show you something here, right? Yeah, you've seen already those slides here, which I prepared. So I've done this already a few hours ago to dry them so that we don't have any waiting time here. Okay, but I'm going to swing over the camera because I bought something. Finally, it's a little bit organized. Yeah, so basically you see here over here, that's my monitor, right? Yeah, you can see. Yeah, you can see me over here on the side. That's where I can read the chat. Yeah, yeah, basically like what you see, but this is basically a little bit new here. I bought myself this here, and to get everything a little bit more organized, this is actually for, how do you call this, for spoons and forks and knives for these eating utensils, right? And I'm just using this also to get a little bit my my microscopy stuff organized. So I've got the pipettes over here, razor blades here. Here's some spare how do you call this? Cover glasses, pipettes and tweezers, scissors. Yeah, some writing material over here. Over here I've got the slides and cover glasses, of course, and here I've got the surface. Yeah, and just also for your information, where you have to be a little bit careful when doing staining. Do you see those blue spots here? That's crystal violet. It's impossible for me to remove, and even with alcohol, it won't go away. Yeah, so I just also wanted to show you a little bit some of the other things here, because when I swing the camera over, right here, this is basically the place where I put the used microscope slides, and here. Yeah, that's my microphone, obviously, and over here in the distance in the back, you see the water samples that basically I'm using. So this one over here, yeah, that's the coffee warmer, and I've been warming again some glycerin gelatin because that's going to be one of the mounting media that we'll be using. Okay, yeah, and the microscope that I'm using is on the other side, and I cannot swing the camera over because it's blocked. But basically, I've got everything here around me. Just wanted to share this with you a little bit so that especially those of you if you're a little bit new to the whole thing, that you know a little bit of yeah, what's going on here. Yeah. So if you have on-topic questions, I'm also going to ask them. I already see the question about nail polish. Nail polish is a mounting medium, has the disadvantage that it shrinks a lot. Okay, so that is a little bit the problem. So what I'm going to do, well, I'm first going to open this here, just for the, yeah, and later on we'll be using the round cover glasses. What I would like to do first is I would like to first show you the samples from last week, because if you remember, last week I mounted some pollen using glycerin gelatin, and there I made 2 pollen slides, and the glycerin gelatin contains some dye. I'm going to now look here. Yeah, that's yeah. Basically, it contains for pollen it contains the dye called fuchsin. And that's why it's so red, and I have to keep it warmed on the coffee warmer. And when you mount the pollen, and I'm going to show this to you now because something has changed, not a lot. See, I have to switch on the camera first. Here we go. Yeah. So I just want to show it to you because some of the pollen grains actually turned. Over here. Here's the pollen, right? Okay. I'm going to now show you. Because now the stain has become a little bit more effective over the last week. You can actually see that the pollen grains now have turned nice and purple because they have now absorbed the stain that was in the mounting medium. Okay, just wanted to share this with you, and I just see that it's kind of a little bit weird because you're not seeing the full picture because there's this weird black, how do you say, frame over here. Give me a second, a little bit. Why is this not? It's a little bit strange. Not this one over here. I have to do, and I'm missing. Yeah, I'm missing the image of the stage, which is a little bit of a pity, because I cannot see the stage anymore. So let me quickly try this. To reactivate this. Yeah, if we're not able to see the stage for whatever reason, I don't know. Folks, I'm sorry about that. But I'm kind of a little bit worried about interrupting the stream. But normally you should be able to see the stage of a microscope up here. And for whatever reason, I'm not able to see that. Okay. Okay. That's the wrong stage. I'm a little bit unhappy. That's the wrong stage. Okay. Yeah, I think there is something a little bit messed up with the USB cameras, unfortunately. But yeah, I'd have to restart the computer. I'm not going to do that now. Okay. That's a little bit of a pity that I don't have this now. But let me try the following. Now, I'm going to improvise now something. I'm going to now improvise something. I've ripped off the camera of my other microscope. I've ripped and let's try if we can get that to work. That's the wrong one. Yeah, okay. See, we're gonna be using this here. It's now a little bit upside down. So, see, that's a little bit one of the unfortunate things here. Yeah, that's, yeah. That should be the stage view. Now with my other camera, because the original camera doesn't work, I'm going to try the following. Sorry, folks, about this. This is a little bit, how do you say, unfortunate. But I'm now trying to adjust everything again. Yeah. So you're seeing basically now the stage from behind, unfortunately. Okay. Yeah, we cannot, unfortunately. Yeah. So this is now, that's basically the question is what magnification is this? Yeah, that would actually be the idea is that you should be able to see the magnification based on the objective. Yeah, so when I change the objective here, but unfortunately I'm not able to put the camera there where it's supposed, where we can see it best. Okay. Now, this is a little bit unfortunate here. But for whatever reason, the camera didn't, yeah. So a new perspective can't hurt. Okay, I'm not going to get myself too much sidetracked here. Just wanted to show you that the pollen grains have now absorbed the dye quite nicely, and this is how it looks like after 1 week. Okay, so this is just something I wanted to share with you. We can go up with the magnification yet further, yeah, and. Yeah, so here, at least the pollen looks still quite nice. Yeah? The thing with the camera is sometimes you see that it will actually switch to black and white. And this is if there is not enough light present, then the camera switches to black and white view. And that's also a little bit, it's actually a feature of the camera, just for your info in case you're kind of wondering. And the original camera, unfortunately, is not working. Yeah? And let me try this again. No, it doesn't even show up anymore, unfortunately. It doesn't show up because I think there has been some issue with the USB connection. Doesn't matter. I'm not going to get myself now sidetracked too much about that. I'm going to switch back now to the desk view. Okay, at least this seems to work. And I'm going to now do the following. We're just going to start observing a little bit so that you know how the samples look in real life. And then I'm going to show you the sample, how it looks when it's dry. And then I'm going to mount it, and then we're going to look at it again. And I don't know if you have enough time for all of this, but that's kind of a little bit the strategy so that you're able to see how a fresh sample and a dry sample and a mounted sample, how they kind of look like, right? And I've essentially prepared 3 samples. The first one is one that you already know. This one contains Daphnia. However, I'm not going to put the Daphnia themselves under the microscope, but rather from the bottom here, on the very bottom, there is some kind of sediment, and I'm going to take a sample from there, and I'm going to, and it contains, all the way from the bottom, and I have to take up some of the solid material here as well, and yep, this was actually already enough, and this contains, a lot of also dead water-free parts. Yeah, diatoms and so forth, and that's basically what I would like to show you first. And if we're lucky, also some copepods, which are also more or less happily swimming around. And those water samples have a disadvantage because generally it's difficult to make permanently mounted slides because they are quite sensitive to drying. Because when it dries, then in many cases the specimens start to deform. I have a little bit too much water here, but I think it should be okay. So we're going to have another view of the scope. Here we go. Okay. And and this is how it looks like. Let's. You also want. Now I knocked the camera. Great. Yeah. Now I knocked the camera. Here. That's me here in the background. Yeah. Otherwise, I'm just not gonna use this camera. Yeah. So this is how it looks like. The water sample. I just have to find. Yeah, here it is. And. Yeah, what you see over here, not only bubbles, but also debris and, here, dead Daphnia, for example, here. Maybe I'm just going to go further down with the magnification because I would like to get a better overview. But this one actually is not super interesting. I just realized I'm going to try this again because I did want to find at least here, there are more parts of the dead Daphnia, but I was actually hoping to find some of the copepods here as well. But maybe because it was already so disturbed. But what we have here is the following. When you actually go up with the magnification, yeah, you see some occasional movement, but this is not specifically a nice sample. Okay, I'm going to try this again because this is rather disappointing. That's one of the typical demonstration things here. Okay, let me try again. So let's try this here. So let's try this here, and I have to make sure that, yeah. Otherwise, I'm just going to show you the sample that, yeah, is already dry. Camera fell off again. Oh, of course. Yeah. And scope. So here we go. Yeah. So what we have over here. Yeah. Yeah. Pretty large. Do you see this here? This thing here, that is actually the exoskeleton of what appears to be of a Daphnia. Yeah. Either because, I don't know, because either it molted or because it died. Yeah. And so especially on the sediment, I find a lot of these parts here, and here another one. Yeah. And I was actually hoping to find some of the because they were just before, but maybe I chased them all already away. There were lots of copepods as well, but maybe because it disturbed everything. Yeah. Yeah. Here again, lots of dead Daphnias. Yeah. Here, this one here again. Yeah. So, and okay, yeah. I think there are indeed slightly more interesting specimens. But what I would like to do now is we're now going to have a look at. I need to wipe the stage. And we're now going to look at over here what I've prepared. Because that is the same specimen, just dried. Okay, and you can see that when you dry it, then of course the thing starts to cluster together and it becomes a little bit intransparent. And what you see over here again are the dead parts of those water fleas and a whole range of other things. Yeah, and the idea is I would like to simply now drop mounting medium on those, and we're going to see how it looks then different. Yeah. So because I've never tried this before. So we go over here. No, I'm not going to do that just yet. I'm going to quickly go through the comments sections first. Wow, lots of comments. I'm just going to clear nail polish. Works, depends a little bit, but it starts to shrink a lot. Yeah. White vinegar on the crystal violet. White vinegar. Leave it to soak for about half an hour and then wash it clean. Okay, thank you for the suggestion. This is basically because of the blue spots that I have over here. Okay, so I'm going to try to use, I've got some white vinegar. It's actually quite a concentrated vinegar, and maybe it does work. So maybe use bleach, could be possible. I'm a little bit worried about using acetone. I was also thinking, because I've got this plastic cover here. Yeah, and so there are some. I just wanted to know. Okay. There are black dots in the center of the pollen. Is this the nucleus? Maybe, I don't know. Okay. That's the thing. I mean, there are so many other things, of course, cell organelles and so on inside the pollen. So one would have to do a specific DNA staining to make sure that it's actually the nucleus. How long could it take to decompose Daphnia shells with bacteria? Is time lapse possible? time lapse, I think, would not be possible so easily, because if you want to make time lapse using decomposition on a microscope slide, then the slide is going to dry up, and so I think that's going to be difficult to maintain a concentrated, a consistent controlled environment for several days. Yeah, so I think watching decomposition under the microscope as such is going to be a difficult thing, because you have to ensure that there are controlled conditions, and I think that might be the more difficult thing. Yeah, so I've got over here I labeled them with crustaceans. So basically these are the shells of those dead Daphnias and whatnot. And over here I'm going to start off with the more difficult specimen, which is the glycerine gelatin. And I have to warm or pre-warm the slide a little bit here, and I'm going to use my pipette, and 50 microliters is the volume that I determined to be fine. Yeah. And I'm just going to give it a try. Let's have a look first here. These are the cover glasses, and a little bit unfortunate always, they seem to be, it seems to be okay, but sometimes they're a little bit dirty as well. Yeah. Hydrogen peroxide can bleach out the color. I've not tried hydrogen peroxide yet, but that's indeed possible. I have got some hydrogen peroxide as well. Yeah? Simply didn't bother yet to try out the different things here. But that's actually maybe another experiment a little bit, is you try to different solvents. So this is glycerine gelatin, which is highly bubbly. Okay, so let's put it on here. Okay, and let's drop the cover glass on top. I have to use, it's easier to use a tweezer. And then let's hope that, it forms a thin, clear layer. And as already mentioned last week, glycerine gelatin basically stays semi-solid. This means that it does not dry completely, and therefore, because it contains a little bit of water, it might not store very long or quite as long, but at least the specimens, apparently, they do not completely deform when the medium dries up. So we're going to give it another look. Where is the scope view? Here it is. Okay. And it's a little bit too bright. Okay. Okay. And I think, here, that's one of those structures from one of those Daphnias, from the exoskeleton, and I think you see that compared to before, it's much brighter. Yeah, it's quite a bit brighter and way more clear. Yeah. So these are parts of the exoskeleton. So maybe we can find something else here. Yeah. Actually, I was kind of hoping to find one of those copepods. Here it is. Here it is. Okay, so that's one of those copepods that was actually swimming around before, and then it dried, and now it's here. Yeah. So, and you see those circles again are those bubbles. Yeah. So that's basically the thing where I'm going to now wait again for several days, and we're going to see whether the pigment, the stain that's included in the mounting medium, whether it actually starts to stain certain structures. Yeah. So again here, this one over here. This one here is, I made a video recently about those. That's a larva of one of those water crustaceans. It's a Nauplius larva. It's not a mite. Yeah, but that is actually the larva of those Daphnias, water crustaceans, and copepods and so on. Yeah, they're called the Nauplius larvae, and they move around quite quickly. Yeah, and here it is. Yeah. Again, we're going to see a little bit how the stain reacts with the specimens that are in here. Yeah. I see this one already, whatever that is, it already starts to turn a little bit purple. Do you see? I don't know what that is in the center. Yeah. Let's turn off the arrow. Yeah, so we're gonna, again here, parts of the exoskeleton. Ah, that's a huge one. Look at this. What is this? I have no idea. Oh, looks almost like an ostracod. Okay. Yeah, this is a big one. Yeah. That was the copepod from before. So that's the first one. I would like to try now. Ah, here's another copepod. Okay. Yeah, and yeah, we're gonna see how this actually reacts with over time. Because I also have UPRL, because UPRL actually has the tendency to clear up the specimens quite a bit. So we're gonna see next week how this actually reacted. So what I still have to do here is I have to label this somehow. This was glycerine gelatin, so I'm going to put a G on here for glycerol jelly. A G, so that I know what this is. Okay, and now I basically put it away. Yeah, over there. So the next one, I don't know, maybe we should do the glycerine gelatin first so that we can move it out of the way. Yeah, then let's do that first, also for a little bit of variation. The second one that I want to show you are nematode worms. And yeah, I've got those vinegar eels in here. It's also one of my more favorite, how do you say, specimen. They look kind of, yeah, extreme. So let me show that to you. They are actually more interesting when they're alive, okay? And when they're moving. But I want to make a permanent mount. Never done a permanent mount of these before. So this is vinegar, 50% vinegar with water, a little bit of sugar. Yeah, and what you see on the bottom here is a piece of apple. And I now have to find, sometimes they will actually swim towards the top. I'm not so sure if I'm able to find a lot of these now here. Okay. So I'll probably have caught a few of them. Okay, here it is. Cover glass. I mean, you've probably already seen them anyway before, but just for reference purposes, yeah, I want to show them to you again. Scope view. Yeah, maybe I didn't catch a lot of them. No. Here's one, right? Okay, yeah. You know, kind of almost like a little snake, or yeah. I mean, they're not. Here's another one. Yeah, because I disturbed the liquid a little bit, so they all went, yeah, escaped downwards. Let's go up. Yeah. So these are nematode worms which can be bought as fish food, and I've been growing them now over a year already in the jar. They're really easy to keep. Yeah, and yeah, you probably yeah, remember one of my videos where I added a little bit of methyl cellulose to slow down their movement. If you don't have that, just use very little liquid, and then it becomes sandwiched between the cover glass and the microscope slide. So this is a little bit yeah, how it looks like here. Yeah, and I'm going to now show you the slide that I've prepared, which contains, which looks very different. There are also more of them on here. And where is it? Here it is. Okay, so that is the slide that has yeah, which is now dry. Yeah, and no cover glass now, and this is how they look like, right? And now I would like to also use glycerine gelatin to make a permanent mount. And then we're just going to try the other two mounting media as well. But I want to first get the glycerol jelly out of the way. Yeah. So I'm just going to quickly go through the questions, just if they're quick to answer. Okay. Otherwise, I'm going to answer the rest later on. It's already half an hour that I've been talking. Gee. How long could it, crystal violet, okay, hydrogen peroxide. Don't use bleach on the melamine plastic desk. It can damage the coating. And what I've got over here, just for your info, this is not directly the desk, but there is kind of a writing surface that I've put on top of it. So the actual desk is luckily not affected. Otherwise, yeah. So that's I put a more or less cheap writing surface on top here. Yeah. So the test patch on this. Yeah. Okay. Don't forget to label the slides. Yes, that's correct. Okay. So just some comments. Could the water in the glycerin affect the microorganisms? Could it cause decomposition? I believe the thing. So there's Tini Coco is asking if when you use glycerol jelly, if this can cause decomposition. The thing is maybe. The thing is that in the glycerin gelatin they included a disinfectant so that it does not turn bad too easily. And normally what you have to do is you have to put also a ring around the slide, the cover glass. So it is correct that every time when you use water-based mounting media which do not completely dry, and there is of course a stability problem, and if bacteria or whatever are able to enter it, then it could indeed decompose it. For this reason, glycerol jelly does not store very long. Yeah. Okay. Will the fuchsine go and the specimen become a bit clearer over time too? I don't know. Marie is asking this question. That is exactly the reason why I'm trying this out. Next week I would like to show you the slides again, and then we're going to see. Yeah. Okay. We can put the glycerin gelatin into the vacuum chamber. That's also correct. Normally you have to degas everything to make sure that there are no bubbles. Yeah. So how do I cultivate the nematodes? Very easy. 50% water, 50% vinegar, a spoon of sugar. Yeah. So yeah. So very quickly only, and now everything here has been pre-warmed, and now I'm going to do this again. And here, yeah, I've already got the nematode slide over here as well. So and I can remove the one from the microscope. And I need another tip. So here it is. And yes, another round cover glass. Yeah, there are plenty of bubbles now. Sometimes the bubbles will pop, and then it will be fine. Let's see if they pop. They should pop, hopefully. Pop. Yes, I think that's okay. Yeah, there's one bubble in the center. It's not entirely centered here. Yeah. So let's have a look now how this actually looks like. And I'm specifically interested in whether it actually is also able to absorb the dye. I tried to stain nematodes with crystal violet, and they do not accept the dye. Yeah, it's kind of interesting as well. Scope. Yeah. Yeah, the camera is now pointing. I don't know. Where's the camera pointing to? So, okay, let's just. The cable is not long enough, unfortunately. Yeah, I'm just gonna leave. You know what I'm gonna do? This is a little bit annoying. I'm going to. Yeah, maybe just take the slide off, and it looks much better. Yeah. I think now they also don't move around quite as much. Obviously not, they're dead. And now you can also observe them better. So let me just quickly get the sources, stage. I'm going to just remove the stage here so we can actually see this a little bit better. Now I have to tell you always the magnification. It's now 10 times. And let's go up with the magnification. This is, by the way, this is bright field. Right? Probably you're always used to a little bit of color. So yeah, if you want to have color, here we go. Yeah? You can choose whatever color you want. Let's add a little bit of color, right? So actually it's much nicer this way to actually observe what's going on inside those nematode worms. So let's now 60 times. Okay, and I have to open it a bit. Yeah, so, and that's basically one of them here. Yeah. So this is, yeah. So as we enjoy this worm here, I'll quickly go through. Jesse's asking, did you get a chance to have a look at the amoeba video? I think, are you referring to the links that essentially you sent me? There were some embedded links. Yes, I got them, if this is the one. Yeah. This is, was linked, and yes, I had a look at them. Thank you very much. Yeah. How do I get your samples? Depends on the sample. Sometimes I go out and collect them myself. What's the color of the glycerin jelly? The color of the glycerin jelly is reddish, purple. Purple not, yeah, a little bit. I just show you again. Here it is. Because the company added a dye called fuchsin, or fuchsin, I don't know how it's properly pronounced. And this is basically glycerin jelly made specifically for pollen. Yeah, there is also another glycerin jelly which does not contain a dye, but this one is for pollen, and usually the one for pollen is glycerin jelly according to Kisser, Kisser, and the regular glycerin jelly is according to Kaiser. And I think the concentration of glycerin is a little bit different in those, right? But this one over here already contains a stain, which is then absorbed over a couple of days. Yeah? So there are different types of glycerin jelly, and as I mentioned already, they are probably one of the more difficult mounting media to be used because you need to warm everything. Yeah? So you mentioned the nematode cultivation recipe, but should there be a starter sample first, like a few nematodes to start? Yes, you need a starter. You don't have that, you can buy them. I got the nematodes from a colleague who's got an aquarium, because those nematode worms are cultivated by people who actually have aquariums. They are making those nematodes at home for live fish food. And yeah, so there should be online plenty of information on that, especially for people who keep a home aquarium. So, oh yeah, you want to see the scope view again, right? So this is essentially, and then they're really easy to keep. They sometimes also suggest that you add a small piece of apple, a spoonful of sugar, and then you basically just have a starter, and then they reproduce like crazy. Yeah. So that is basically the glycerin jelly. And the nematode worms, and then I don't know, when you take away the filter, then maybe over time, I don't know, maybe they're going to turn also reddish. I don't know whether they're going to accept the dye or not. So the last one, is one with let me label this first. Where's the desk here? So this one here is the nematode worms with, and I put a G on here for the glycerin jelly. And the last one that I have, and then I'm going to mount, of course, also using the other mounting media. Oh, I always have to keep this warm. By the way, when you're using glycerin jelly, be careful, don't overheat. It's really important. If you make it too hot, like in a hot boiling water bath, then the glycerol jelly might not turn solid anymore because the gelatin breaks, right? So I basically warmed it only to 40 degrees, and this took quite a bit of time So the next one is the following. Which one did I use over here? Is simply a water sample that I collected last week. In here. So, and put this away. You see, these slides are dirty. I washed them because I'm recycling all of the glass slides, but not all of them are actually very clean. So, and here I need my tweezers, which are lost again. Where did I put them? Here. Here they are. And what I do is simply take a little bit of this green stuff here, and that's it. That's basically what I've done also before. The rest goes back. Cover glass on top. Where is the cover glass? Yeah, one of the things that I would like to have here is slightly more space. So cover glass goes on top. I always make sure that there's only one of them. It's still not a lot of liquid, but let's have now a look how the original sample looks like. I need to go to the scope view again. Yes, that's the diatom one, right? So this is actually quite a nice one because there are diatoms in here. Now diatoms have a silica shell, so it should not be such a huge problem. Them losing their shape. Some of the diatoms actually are gliding around. Again, now this is bright field, so you see the natural colors here, right? There's some other organisms there as well. Yeah, but these diatoms are some gliding diatoms that you have here. Yeah? So I've got a lot of questions today. This is probably the last. How do nematodes have enough energy to move so much? As a matter of fact, I don't know if you watched my video some weeks ago. I actually made a video about the movement of the in this channel. Basically, why are they moving around so much, right? What's the whole point? Is this not kind of a waste of energy? Just please check this channel. A couple of weeks ago, I actually made a video on this. And the thing is of course, it requires a lot of energy, but that's why they are absorbing a lot of food also over their skin, and they also have a digestive system and so on. So this is basically their way of also obtaining food, and also by moving around so much, they're also mixing the water a little bit, and this is also a way of how they can get more oxygen, right? So this is different organisms have different strategies, so to say, to solve different problems. It's here, especially the higher market. This is actually, I think that's a nicer, also quite a nice sample. So again, I made a how do you say a slide where I dried. So let's remove this again. So that's the fresh one, and this is the lake sample. Let's put this one over here. And that's the one. Basically, sometimes, this is the one that essentially is now air dried, and you probably notice that it looks a little bit darker, and the reason is that's very often the case because the refractive index of the object to the surrounding air is significantly larger. This basically makes them appear a little bit darker, right? As always, let's try to now make a mount using this here. Okay, so let's remove it again. And the desk view here, and then that's going to be the last. That's going to be now the last one that I'm making today with the glycerine jelly. I don't know if this is still able to. I think it might be clogged. So. Always close everything so that in case you knock it over that it So let's give it a try again. Spreads nicely. Let's hope that the bubble pops. Should pop up. It's a little bit, maybe it wasn't quite enough mounting medium. It doesn't matter. I just wanted to show you, and there is now a gigantic air bubble here in the middle. It doesn't matter. It's just for testing purposes anyway. And here we go again. Where's the scope view? Here we go again. You see that, lots of bubbles here. Let me see if there's, This is how it looks like. Some bubbles are floating around still. But still somehow not quite as bright as the original one. It's a question of refractive index. Let's go up with the magnification again. Yeah. And also here, I'd like to know a little bit, or I'd like to see how the dye that's included reacts with the specimen. If there are certain algae or so that will turn pink or not. Yeah. So I will quickly have a look here. Okay. This is one of the first surprising things I've learned using a microscope. Algae and diatoms are moving organisms, not only bacteria. Yep. Movement is actually a very common thing. It's one of the characteristics of life. And even if the organisms themselves don't move, then at least inside the cell, you have sometimes or often quite a bit of movement. Yeah. Off topic, what was discovered about bacteria before DNA analysis existed and how was that done? Oh my gosh. You know what? Thank you for the question. So this is a question about DNA analysis before, how were bacteria characterized before DNA analysis? You know what I'm going to do now? I'm going to, thank you for this is a huge question. When I studied microbiology, I still did classical microbiology. So my job is to basically classify and identify bacteria based on biochemical tests. So the short answer is you can grow bacteria, you can purify them, and you can do chemical analyses on the bacteria. And depending on what chemical compounds you find, you can characterize bacteria this way. It's called chemotaxonomy. Yeah, that's basically something that I've been doing during my diploma thesis many years ago. So you can, there are a variety of ways how you can do that. You can also do metabolic tests if they're able to break down certain substances or not. So there are a variety of different ways how you can do that, and it depends really a lot on the questions that you want to answer. Right. So yeah, if it's for medicine or if you just want to characterize them in very much detail and so on. Yeah. How long does it take before you can apply the enclosing ring, please? Honestly, that's I don't know exactly how long it takes, because normally what it says is you should let it cool out a little bit, the slide, and then it's going to dry a little bit, and then you have an enclosing ring. But I would say, I have to admit, I have not found any any instructions yet. But you know what I want to show you? I want to show you two books, which are already, let me quickly put I accidentally made this a little too hot now. Yeah. So I'm going to show you now two books, and basically I would like to, I'm in the process of reading them now, and this one was recommended by one of you, Safe Microscopic Techniques for Amateurs, and this one I bought also from Brunel Microscopes, and I've not read it yet. Okay, but this one outlines how to use glycerine jelly, and then maybe I'll find somewhere information on how long you should allow the medium to dry before you make a ring around it. Okay, yeah, yep. I've just seen 55 is still okay. What I read, right? And luckily it wasn't that hot. You should not go above 55, however. So, but what I read, 55 is still okay. So what I'm going to do now is because otherwise we're running out of time, I'm going to be now a little bit more efficient, and I'm going to now do the crustaceans, lake, and the nematodes. Okay, and I'm going to do these here now a little bit faster. Okay, so I'm going to use now Uprol very briefly. Why this medium, strictly speaking, might not be the best medium. I've used it quite a bit, but generally because it contains a solvent, and therefore anything that is pigmented, like for example the green algae and so on, it will dissolve the chlorophyll, so that there can actually be some kind of a bleaching out. But that's exactly what I would like to test, right? So I'm going to quickly try this. I'm going to add a drop on each one of them. I'm going to make this very efficient. I'm going to go one by one. It takes approximately 6 weeks for UPROL to dry, and during that time you have to store the slide, of course, horizontally. Okay, yeah, it smells nice. Yeah, UPROL is a medium that is essentially commonly used by people who are mounting insects and other arthropods because it has a natural clearing action. So the exoskeleton, even of dark objects, dark insects, becomes clear. See, this one was not quite centered. Yeah. So number one, let capillary action do its rest. You can also speed up the drying time of UPROL by actually warming the slide. It's also sometimes done. See, unfortunately, I've spilled out now a little bit of the medium here on the side. That's okay. I just want to also give you a very brief warning. The medium is not dry yet, and you have to be careful that you do not touch the side of the microscope objective. Okay, so I'll be only using the low power. Okay, I'll be only to make sure that there is enough distance. Okay, so that's the UPROL one. That is actually an easy one to identify because the medium is a little bit yellow. Okay, so we're gonna give it another try. So this one over here was, what was this? That's the lake one. I have to also label this first with the glycerine jelly. So let's have a look at the nematode worms. Again, the mounting medium always should be compatible with the specimen. And then I'm going to have a look again at some of your questions. So here we go. Oh, there are lots of them here. Bright field again. Let's add a little bit of color. Okay. And usually it takes a little bit some time, and then we're going to see whether by next week those worms have somehow deformed a little bit, shriveled up, shrunk up, whatever, right? Because of the solvent. Okay, so that's basically the nematode worms. Yeah. So the next one are those hopefully those water crustaceans, where we were able to find some of those how do you say this, copepods and so on. I don't know if there are anyone. Yeah, that's basically the one from containing the dead exoskeletons. So to quickly go through the whole specimen, maybe we're able to find it might be something here. I've briefly checked before, and there were a couple of copepods there, but I don't know if I'm able to find them now. This one, of all of the 3 specimens, this is not the most interesting one. I just realized. So yeah, bubble. Yeah, and this one over here again. The exoskeleton of a dead Daphnia. Yeah, this is not a particularly good one. Yeah, let's move on. And the last one, if I find something, then I will, how do you say, remember the position, and then I'll try to show you next time. This one here is the lake, the one with the diatoms. So, and this is how it looks like, okay? And yeah, it doesn't, so we go up with, so we're now at 20 times the 20 times objective. Yeah. And we're going to see that sometimes there are some air bubbles inside the diatoms as well. Yeah, and we're going to again have a look on how well this actually, let me put the filter out. By next week. But we can already see it. That's what I actually wanted to show you. Okay, actually, I don't know if you should be able to see this as well, but do you actually see that the green pigment is starting to be washed out? Look, I don't know if the arrow is now a little too large. But there is this, yeah, I'm not in bright field, but you see there, it's starting to become a little bit greenish, yeah, around the diatoms. And this actually means that the chlorophyll is being dissolved by the solvent of the mounting medium. Yeah. So this is a little bit of the thing why you should use for these algae and so on, why you should use a water-based mounting medium, because yeah, it dissolves the chlorophyll just like I predicted. Yeah. I can see this here really well. I hope that this color information is not lost over YouTube. Yeah, but here you should also be able to see that the background starts to become a little bit greenish. Yeah. Yeah. So I'm reading it again. So I'm going to now have a look again at some of the comments here. How long does it take? I don't know. Can you have a morning stream for other people easier to watch? Thinking about it. I'm thinking about this. I know that some people requested also a morning live stream. I have to tell you, it's going to be a little bit difficult to kind of combine that with my work. Well, it could be on a Saturday morning or on a Sunday morning. If I were to do that, 2 live streams, then I would probably make 2 shorter ones. Now, I don't know. I'm doing the live streams for 1.5 hours. I don't know if this, yeah, is too long or too short for you. Yeah. So that is still, yeah. I'm welcoming any type of feedback. Yeah. I'm skipping over some of the comments and I'm going to answer them later. Yeah. There is a mounting medium called Entelan. I know Entelan, the mounting medium by name, but I have not used it. Generally, many of those synthetic mounting media require xylene as a solvent, and I try to avoid that. Yeah. Yeah. So I hear that basically I'm getting now the questions. Thank you very much. Thank you. I got your questions now, which are a little bit off topic, and I will try to answer them as well. 2 pages again. Wow. Okay. But we're not finished yet. I will do first those here, the remaining. But that is actually something I wanted to show you here, is that it's starting to, as you can see, it's washing out the green pigment. Yeah. For this reason, this medium is not very suitable for that. So, and the last one is also one that you already know. Where is the desk view? This one over here. This is clear Elmer's PVA glue. I'm sure that there are also other companies. If you're in the German-speaking world, there is also, I have not tried it yet, but maybe you can try it for me. The glue called Uhu without solvent. Uhu is the name of the glue, usually contains acetone as a solvent, but there is also one that's free of solvent. And it's water-based, which has a very similar characteristics. And unfortunately, Elmer's PVA glue was very cheap in a large amount, but I had problems getting it because most of the PVA glues that they're selling these days are white. Yeah, but this one is actually one of this was clear. And not only that, I diluted it down with water. Yeah, so I added a little bit of water so that it's easier to use in a pipette. Bubble. So yeah, again, the whole thing with the bubbles is an issue, but the idea here is, I just want to demonstrate this to you. Yeah, so this is, again, the reason, I know I repeat myself, but I think that's the choice of the mounting medium. That's why there's so many different mounting media around, is because of the refractive index and also whether the solvent is compatible with the specimen, right? So here, that's the one over here. The next one is over here. And I've actually seen sometimes that some mounting media simply also had the wrong refractive index and it didn't look good at all. And the last one. I'm doing this very quick now. And some of you might have also heard of the medium Canada balsam, which is a traditional one, which essentially, if properly prepared, apparently the slides, they last an eternity. But they have to be completely, specimen has to be completely dry. Yeah? So that is now the PVA glue, and let's give it a try. Here again in the scope view. Here again the nematode worms. Okay. You know what I'm gonna do? Why not try the following: nematode here, nematode here, nematode here. Let's try this one over here. That is now the. Let's try the two water-based mounting media. So that is over here the 20 times bright field. Okay, PVA glue, and over here, maybe it doesn't make a huge difference. And I need to find something here. Here. And that's the glycerine jelly. Yeah, glycerine jelly. PVA. I do not see a huge difference. Okay, but we're gonna wait until it dries. But so far the glycerine jelly, the nematode worms did not absorb the pigment. Okay, so that's also something noteworthy. So and over here quickly the nematode worms using Uprol, which contains the solvent. Ha! Ah yeah, look. It doesn't look quite as nice as the other one, right? Yeah, it looks kind of a little bit shriveled up and yeah. Yeah, so you can see that the Uprol as a mounting medium is not quite as suitable here. They don't look quite as nice, right? Let's compare it again with the PVA, the water-based one. Yeah, the worms look much nicer. Let's try just for the fun of it with a little bit of color. Yeah. So that's the PVA glue, and over here this is interesting. That's the one now with the Uprol. The worms kind of look a little bit flatter, almost like they've of course maybe they were kind of the solvent might have kind of removed the water a little bit. Yeah. But it looks a little bit like there's something in it now. Yeah. So yeah, just wanted to show this for comparison. And what else do I have? Yeah, the last one. So these were the worms. And I have to label it. Just a second. Let me put a PVA written on it. And over here, this is now that's the boring sample, the boring one, the one from. So that is, let's go bright field again. No, wrong, that was the wrong filter. Here we go. What is this? Some algae, okay. Here, that's another Copic pod. This lots of big air bubble, but maybe more interest again the different parts of the dead water crustaceans here, but maybe we're gonna have a look at the ones with the diatoms. And that is the PVA glue with the diatoms. And you know what I'm going to do now is I'm going to put the diatom one and the one with the lake with glycerin jelly next to each other. Let's compare the two water-based mounting media again. So that is the glycerin jelly. Okay, the bubbles. And that is the PVA glue. I think it kind of looks similar. Yeah. But at least there was no bleeding out of the colors. But we're just going to wait for a couple of days, and we're going to see how this then actually compares. Okay. So, what I'm going to do now, what time is it? Ooh, okay, yeah, I'm going to give you again a desk view. Over here are the questions. Let me quickly see. Ooh, I love it. There are lots of comments. Okay. I think the UPRL slide, yeah, you know what I'm going to do is I'm going to go through the questions here first, okay? And then I'm going to have a last look again at your chat, okay? Just a second. Are these the right questions? Just a second. Are these the questions that I think I'm a little bit concerned. I think my little helper, my son, printed out the wrong questions. I'm going to just a second. These were the questions of last week. Yeah, just a second. Michael, du hast die Fragen von letzter Woche ausgedruckt. So I'm back. Yeah, my son unfortunately printed out the questions from last week. Yeah, so that's why I asked him to print out the new ones. Yeah, this can happen. He's. By the way, I'm going to use this time now for another little advertisement. Not an advertisement, because I'm receiving so many questions, and because I want to kind of think of always of new things to keep the channel active. Once a week, I'm now also uploading a, how do you call this, an audio only podcast. Where I basically just have myself talking and answering questions. So if you go into and click the podcast tab in YouTube, then you should be able to see, and also in the videos, you should actually see that last week I uploaded the first one. Tomorrow I'm going to upload another one. So usually for about 30 minutes, I just basically want to how do you say, just talk about a variety of topics and answer your questions. Yeah. So this is a little bit something that I'm also trying to experiment right now. Yeah. I myself like to listen to podcasts when I'm driving to work, and so I thought maybe, and I do get a lot of questions as well that I would like to talk about, but they do not need some kind of microscopic video. So it's kind of like a talking head type of thing. You might want to check it out. I've tried it last week, the first episode. And yeah, it will be just one of the normal YouTube videos, but you can also treat it like a podcast. Yeah. So you know what I'm going to use this time here? What are protists? Oma is asking. Protists are single-celled eukaryotes. Eukaryotes are those cells that have a nucleus with a DNA, and protists are also known as protozoans. Are single-celled. The most famous one maybe is the one that causes malaria. Yeah. So the pathogen that causes malaria is a protist. Yeah. Could you upload the podcast to Spotify? It's on YouTube Music. I think it should be there as well. I mean, what I've done is I had a separate podcast also over the webpage where I simply uploaded the MP3 files. But now, because YouTube also allows podcasts, so there are over 30 episodes already. But if you, I put them all into the podcasts playlist in this YouTube channel. So you should find them all. Should find all of them. So they go back a couple of years. I've been neglecting the podcast a little bit. Then for some time, just have a look at it. Maybe it interests you. Yeah. Yeah, this is Jesse's. Thank you here. The podcast was great. Please always keep a video version of it available. I need the visual even if it's just talking. Otherwise, yes, that's actually one of the reasons why I actually have this talking head type of thing, because this non-verbal communication, when you're actually seeing somebody talk, it actually makes it easier sometimes to follow. So this is the reason why I actually decided to not only do an audio only version, but actually to do a talking head type of thing. May I suggest to do a quick publicity to invite people to your previous video and forum? Thank you very much. Here are the questions. Yes, I do have a forum as well. I just need to say the following. Over the last couple of months and years, the forum was spammed by automatic registrations. So if you want to join the forum, then please sign up for it. But you have to send me an email with your username so that basically, I can unlock you. Yeah, I'll do this then very quickly. But there have been some big complaints, justified complaints, because there were so many spam registrations, and with the AI system, there's a question of why do you want to join the forum, but with all of this AI things going on right now, it was impossible for me to actually distinguish if the registrations are by real humans or not. So if you want to join the forum, then please sign up first, and then send me an email. Yeah. So this is a little bit the thing. Yeah. Okay, so what I'm going to do is I'm quickly going to go through the questions here. Wow, this is a lot. I'm happy. I don't have ovaries, okay, or glycerine, but can I use nail polish or other alternative? Okay. Yeah, you can use nail polish. I've also tried nail polish. I will tell you the problem with nail polish is as I mentioned, it shrinks a lot, and it contains also a solvent. So if you want to use nail polish, do the following: allow it to dry first, and make it a little bit less liquid so that it does not shrink quite as much. But I've heard of cases where actually nail polish was actually used also in research. Because it had just the right refractive index and everything. Yeah, and for this reason, I know of someone and they have actually used nail polish, I think, to mount testate amoebae or something like that, if I remember darkly. So it is actually, nail polish can be used and is used. However, it does shrink a lot. Yeah. White vinegar, yes, we talked about this. This refers to the my surface here, which basically contains the stain. Okay, we've talked about this bleach, yeah, crystal violet. Okay, what other stains work well with pollen? I want to make permanent pollen slides, or maybe use no stain. Generally, my suggestion is, what? There's another page. Okay, what about this here? The last page has to be exchanged. Thank you. It's fine. Okay. Yeah, my general suggestion is you don't have to stain it, right? Yeah, you can use normal. Sometimes if you just use stains like I've tried, I think also crystal violet. Indeed, you can try to use fountain pen ink and so on. Yeah, but generally, my suggestion is try it out first. Yeah. So I want, no matter where I look on Google, thank you very much. There are black dots in the center of the stained pollen. It could be the nucleus. I don't know. It could be also some other cell organelles. How long would it take to decompose Daphnia shells with bacteria? Is time lapse possible? No, I don't think it's possible, because that would be under a microscope slide where you would decompose it, right? So if you want to just get the shells, I would suggest that you try treating them with hydrogen peroxide. I've not tried it on Daphnia yet, but I've used hydrogen peroxide on insects, and it bleaches them as well. Yeah, is crystal violet an organic compound and hydrogen peroxide? Yes, because hydrogen peroxide oxidizes organic substances. Yeah. So, you know what, I might actually try that. Yeah. Don't use bleach. Okay, to label the slides, I've done this. I've made a Daphnia swimming pool by placing waterproof tape with a central hole made with a paper punch. I then made several layers of thickness with a central hole, cover slip on top. Ah, okay. You took waterproof tape with a central hole, and then you used a hole punch to make basically a little, spacer rings, so to say. That's an interesting one. Okay. Or, another thing, I don't know if this works, maybe some kind of an old credit card or something like this, a plastic card. Maybe that would also work. Yeah. Could the water in the glycerin affect the microorganisms? Could it cause decomposition? The thing is the following. Glycerin, I already mentioned this, the glycerin gelatin contains a disinfectant. And as a matter of fact, it's also the glycerin which actually is osmotically active. This means that it actually attracts water as well. So it dries out the organisms a little bit, the presence of the glycerin. Yeah. So I think the glycerin as well and the disinfectant that's added both prevent decomposition from happening. However, what I heard is that you have to be you still have to wring the slide to make it more stable. Yeah. Will the fuchsine go and the specimen become a bit clear over time too? I don't know about that, if the fuchsine will decompose, but generally many stains in nature, essentially they don't hold forever, because stains which absorb light, that's a form of energy, right? And it could be that some stains actually will decompose and break down. But I don't know so much about the stability of fuchsine. Yeah. If you don't wait too long, you could put the glycerin gelatin preparations into vacuum chamber. Yes, that's also possible. Yeah, I see. And I think you've also made last year or so. I think if I remember, you presented a vacuum chamber, right? A homemade one. Yeah. How do you cultivate nematodes using vinegar? Yeah, quite easy. Half vinegar, half water, so it's not too concentrated. Spoonful of sugar, a little piece of apple for additional nutrients. Yeah? What are you doing on today's live? Well, I was mounting some slides. There are 4 main types of nematodes in soil. Beneficial ones, root feeders and fungi feeders. Use high magnification to look at the difference at the mouth. Ah, that's an interesting one. Okay, so depending on the type of food that they eat, they have a different, apparently different mouth parts. Yeah. Yes, I had a look. Thank you. You sent me the links. How do I get my samples? My suggestion is that you look at the video that I just recently uploaded. I made one on a snail, and the one before where I actually went out a few days ago, I think it was on Tuesday or so, to collect a water sample. What I do is I just basically go out. I have those plastic tubes, these are centrifuging tubes, and I simply collect samples in here. And that's it. Yeah. So it's really easy. And then basically you have to be a little bit lucky as well. Yeah. Okay, what's the color? I've talked about that. It's slightly reddish, so I already answered this question before. You mentioned the nematode cultivation recipe, but should there be a starter kit? Yes, that's also something. You have to indeed get a starter kit under quotation marks. They're being sold, they're called vinegar eels, and are being sold in aquarium shops. And a lot of people who have a home aquarium actually will breed them as live fish food. Yeah. How do you clean slides for reuse? Can cover glasses be cleaned as well, as they are very brittle? The answer is I basically clean all of my slides, because I generally do not put any, infectious material on the slides. I rinse them in hot water, and sometimes that hot water is enough, sometimes a little bit of soap. And then at the end, you have to rinse them properly and dry them properly, otherwise you get water spots. I also try to clean the cover glasses, but indeed many of them break. Yeah, so I would say you just have to be careful, otherwise you kind of hurt yourself a little bit. So I just, because you're asking, because I reuse the slides, what I've done is, and you see that not all of them I've cleaned really well. There are some water spots on here, but I've deliberately buying now slides that have basically over here edges that have 45 degrees. So, and also over here, they're also not quite as sharp, because they're also polished over here. So there is less chipping, and the reason why I'm doing that is because I don't want to hurt myself when I wash the slides. Yeah. So I decided to buy, because I'm recycling them and reusing them, I decided to pay a little bit more into slightly higher quality slides, and then I'll reuse them a few times. And if they're too dirty and cannot be properly cleaned, then I just throw them out. Yeah, but it's basically, and I've completely phased out the other slides now that have sharp corners. Yeah? Otherwise, when you wash it, and then you might actually hurt yourself. Yeah? So, but different people have different things. The big blob thing I found was strange to find. I got a time lapse of it eating, which I will also upload at some point. Ah, okay. You found, I suppose you're talking about the slime mold. Did you get the samples from a puddle in a park or something? Yes, all over the place. Please watch my previous video where I basically, I think the thumbnail I called Microbes Inside. right? And it actually is a little bit of a vlog style type of video where you can actually see me collect samples. Yeah, and it's really, it took me about 10 minutes or 15 minutes. It was quite cold, but it just, yeah. It could be nice to show a schematic with the 4 different mouth structures of nematodes. Ah, okay. You want to have a little bit of theory. Okay. So because the different nematodes eat different things, yeah, and they have different mouth parts. I've got a lot of questions today. It's probably the last. Do nematodes have enough energy to move so much? Yeah, I also briefly addressed this. They will, of course, it needs a lot of energy, but yeah, they will, of course, feed, and that's why you put them into vinegar with sugar so that they have enough energy. Yeah. The worms you were talking about being in an aquarium are called detritus worms. Yeah, detritus worms is a general term because these detritus is basically anything biological, organic that decomposes. It's called they're full. Yeah, worms love to eat this stuff. Yeah. This could be a nice Mikado game for very precise people. Okay, this is one of the first surprising things I've learned using a microscope. Algae and diatoms are moving. Yes, not only bacteria. Yes, the coffee heater was a little too hot. What was discovered about bacteria before? You know what? I think this is worthy of a podcast. Okay, it might be a little bit of a slightly longer one. Yeah, either a video or a podcast. I'll think about it. Yeah. How long does it take before you can apply the enclosing ring? I don't know that. This is I have to read in the book, and I've not so far. It didn't really mention any time because usually what happens is that after the glycerine gelatin cools out, it becomes solid, and then you should be able to enclose it. Yeah, because it doesn't really dry out. Yeah, I was trying to gather some specimens from the ocean water, but I was able to discover only diatoms for some reason. Yes. Depends where you look, I guess. So my suggestion is always try to scratch something off a surface, of a rock, for example. Yeah. Can you have a morning stream? Yeah, that's still something organization-wise that I have to think about. Yeah. I don't know how many people would join in on the morning. Yeah, I mean, of course, there are time differences all over the place, so they're different in different continents. Yeah. Book recommendation, Animal Life in Freshwater. Thank you. I'm always happy for book recommendations. Yeah. So in April, we'll visit these slides again. Yeah, 6 weeks. What I will do is maybe I'm going to show them already next week, but it indeed takes the Uprol several weeks to actually dry. It's pretty long, yeah. And you have to store it horizontally. Yeah, it's not good to inhale Uprol fumes. I know, but they smell nice. Yeah, it's not as unsafe as xylene and for some others, but you should still take care with it. Here is the full safety data sheet. Okay, thank you. Yes, Uprol indeed, it contains a solvent. So therefore it's irritating. Yeah, and you can use Uprol essence to dilute it down, or you can also use alcohol to dilute it down. Yeah, but. Yeah, it's always something where you have to always be careful when using fumes, mounting media that contain solvents because of the fumes. Yeah, I use a mini soldering hot plate for chemical melts for polarization. I imagine it would work well for warming the jelly. Aha, okay. Yeah, mini soldering hot plate, interesting. Just be careful that because the glycerine jelly really breaks down when it's too hot. So it should not become too hot. So if you're able to adjust the temperature, then it should be not a problem. Yeah. Except for the bigger species, your best chance for extracting some of the target species for mounting would be a great concentration in its favorite medium. Yeah, I don't know. Your best chance for extracting one target species for mounting would be a great concentration in its favor. So basically, the medium that's surrounding it, the mounting medium should be ideally not too different. I don't know. This kind of is difficult from the actual how shall I put that? You see, during the processing of the specimen, often you have to dehydrate it, and usually you're starting to introduce artifacts this way. So this a proper processing of the specimen probably is also very important, and if essentially the medium also has to be compatible, as we've seen, otherwise it starts to bleach out. Yeah. What's the best way to minimize bubbles? Yeah, you could degas it. That's one way. A little bit of a trial and error and a little bit of luck. Okay, and so that's what yeah. And the glycerin jelly, you're not allowed to shake and move too much, otherwise you're introducing bubbles. Yeah, named Entelan. Yes, I know it from by name, but I've not used it. Yeah. So what I'm going to do now is the following. I'm going to quickly go through some of the comments here very quickly, because maybe something there are a couple of comments, and I'm going to go do this quickly. Ooh, there are lots of comments. Entelan, except for, yeah, okay. Air bubbles, vacuum chamber. I suggest you use a yellow light or overlay so it shows microbe organelles in more detail. I don't know why it works, but it works at least on my microscope. Yeah. Okay. So basically, I can imagine. I also don't know why. I mean, it depends a little bit on the pigment of the organelles. If you're using color filters, and it can indeed be that. Because of if they're complementary colors, then some of the organelles might show up a little bit in higher contrast. Yeah. Had a go at a thin section of a sea urchin spine, moderately successful, but still working on it. Please, Dave Lewis commented that he's trying to actually make a preparation of a sea urchin spine. If you actually have figured out how it's done properly, please, I would be very happy for some information, because kind of polishing it down, I was thinking maybe you have to use some epoxy glue to mount the sea urchin spine so that you can polish it down. But if you have any recommendations, then please do share them. Bioluminescence, like in I don't have dinoflagellates. That's a little bit of a problem, so I'm not able. Bioluminescence would be interesting, but I don't always have all of those specimens available. Yeah. What are chloroplasts made of? Proteins? Do they contain DNA as well? Yes. That's a huge question. I mean, chloroplasts have their own DNA. They have a double membrane on the outside. They have thylakoid membranes on the inside. The biochemistry of chloroplasts is fascinating. Essentially what they're made of, the same components as the rest of the cell. It's the structure that's, of course, different. Yeah? So it has phospholipids in the membrane, proteins, enzymes, all these things. Yeah? Water, of course. Yeah? So I would suggest only to keep one live stream a week if you want to keep a happy marriage relationship. Yeah. So quickly go down here again. I accidentally ordered the white PVA glue, and it is very annoying to pipette, and the slides are very ugly. Is there a way to still make it work, or should I throw it out? I don't know about the white one. Honestly, my philosophy is the following. I mean, my suggestion try to thin it down with water. My suggestion is, or that's at least what I try to do as a philosophy, try it out. If it doesn't work, right? Try to optimize it a bit, but if it doesn't work, throw it out. There is no point in trying to make things work that probably won't work. I mean, it depends a little bit what your interests are. I deliberately try to place my focus mostly on nature observation. And there are probably many glues out there, or many things that could work as a mounting medium. But if it doesn't work right away, or after a little bit of tweaking and tuning, then I simply say move on, right? But it depends where your interests are. But I think that's a little bit my philosophy, because otherwise you're going to spend so much time trying to optimize it, and then maybe it won't be successful. Yeah. I think the U-Pearl slide made the nematode organs more visible. Yep. So comment. The podcast was great. Please always keep a video version of it available. Yes, I think I already read that. Yeah. Yes, the forum thing here. Off topic, Bill from Kentucky, USA. Would you consider doing a live stream on soil analysis? Many farmers use several fields around here, plus take soil out of forest to analyze. I will be very, yes, just some interesting background info. A couple of years ago, a company, this was 3-4 years ago, a company in Germany actually sent me different soil samples. This was a composting company, and they requested whether I could make some videos on certain microorganisms that are found in the soil for promotional reasons, right? Because they wanted to have some pictures for the webpage. So they sent me all sorts of different types of composts. Yeah. And I've essentially done that, but honestly, there are, of course, many bacteria in there, some fungi and so on, but you don't see a lot of, how do you say, interesting movement going on so much, right? So what I had to do is to make something interesting. I had to actually enrich the culture. So I extracted the soil sample, I added some food, and I got basically I multiplied the organisms so that there was something there to actually observe, which is sufficiently interesting, right? So that is certainly something that one could do. The question is if you take out soil and if you look at the microbes, that's fun to do, but what does it really tell you? So that's a little bit the question. I mean, it is interesting to look at. But that's pretty much it. If you say, okay, you want to know which type of organisms are there or how many of them are there, then this really goes complicated very quickly. Yeah. So for observation, for exploring the environment and soil, that's certainly possible, but not for actually analyzing it to determine which soil is now healthier or better or contains more microorganisms. Yeah, so that is to do these analyses. It would be quite elaborate, I would say. Yeah. Yeah, soil and not sure why my phone cut it off. I think they mix 1 milliliter of soil and mix it with 6 or 7 milliliters of water. Yeah, so basically what you do is you mix it, you swirl it, and you look at the liquid. Yeah, so and I found a lot of tested amoeba, a lot of fungi. Yeah, and so on. Yeah, yeah, could also be an insect egg larva identification and plant disease from garden. Yeah, that's. But if you look at the leaves under the stereo microscope, that would be possible. Yeah. A slightly less common water microorganisms. Okay. What I generally found out is that when you collect water samples, like I've done over here, and if you let it stand at room temperature for a week or so, what generally happens is because of the change in oxygen concentration and also temperature, what you have is you have some kind of a progression of different microorganisms. So what I found the highest biodiversity if you just go directly and observe a fresh water sample from a pond. And take it home, the sample, and observe it and make videos immediately. How do you collect and extract microbes from soil? Okay, soil microscopy seems to be an interest. I'll take that up and I'll try to do a video on that as well. Okay, basically you mix the soil with water and you you filter it off so that essentially the soil particles are not there, and you observe what you find. Yeah. So on that subject, any chance of a future video with other species of slime molds? From what I've read online, some are made of fruiting bodies. Yes, actually they do. I'd have to get some slime molds, honestly. Okay. And I quickly go down here. The colorful ridged sea urchin spines you have shown before with the bright red and blue colors. Where did you acquire them? I've not managed to find them to buy anywhere online. I got some of the slides are antique slides which I bought over eBay. Yeah, and it's a question of luck. Yeah, so yeah, So that's I quickly go down here. In soil samples, you mainly have nematode identification, fungi beneficial or not, humic acid cluster, anaerobic fauna. Okay, yeah. So I read which liquid is proper to clean an oil from an oil lens. Generally, what you do is if you have oil, you use tissue paper and you only use alcohol. Okay. There are also online tutorials on how to clean the lens, but you have to be a little bit careful that no alcohol goes into the lens. And generally you should not, if you use oil immersion lens, there is no need to wipe it off always with alcohol. Yeah. So this is simply something that I also would like to say. Cleaning of oil immersion lenses is only necessary, if it's really badly contaminated, I would say. So this was a lot of questions. Thank you again. So for those of you who just, I quickly, as a last item, I just quickly summarize what we've done today. Okay. So I hope that I have got all of them. That's UPRL. Oh yeah, that's PVA. Yeah. So here are the slides, and what I'm going to do then is here the remaining slides. Okay, so here they are. This one is UPRL. This one is UPRL. I forgot to label the UPRL ones. Yeah, but they're easy to see because why are they so easy to see? Because they're yellow, so I need to label them here as well. Okay, and that's PVA, and that's also PVA. Yes, luckily I did not label them, but I can actually see it because they're yellow. Okay, and so that is basically what I've done today. What I'm going to now do is I think I'm just going to call it a day today. We're going to have a look at them next week again. Please, I just wanted to give you also a little forewarning. Maybe next week I won't be able to do it on Saturday. I might, but I'm not sure yet. I might have to move the live stream to Sunday. I don't know yet. I might have an appointment on Saturday evening. So please do check the announcement of the live stream. There's always this countdown timer so that, essentially you know when it actually starts. Okay. So I'm going to do the following. I'm going to, what I will be doing is I will be checking some of your questions again, and if there are some questions that need to be a little bit more explanation, I'm going to pick them up also in this weekly, how do you call this, podcast that I'm trying to do now, because why not? Okay, for me it doesn't take so much time to actually simply answer those questions, and please have a look at it, and I'm happy for any feedback that you might give me. Okay. I know I could not answer all of the questions in detail, but for today, I'm going to say I wish you all the best. Thank you that you joined me again. And I wish you a nice weekend, remaining weekend. Happy microbe hunting as always, and see you again in a couple of days. Bye-bye.